The last thing that I want to cover today is microbial metabolism. Okay? Whenever you hear the word metabolism, what metabolism means is all of the chemical processes in a cell or organism. Okay, so that means both building things up and breaking things down. I emphasize that it's both because a lot of students, when they hear metabolism, they just think of their own body and how people commonly use the phrase, oh, I have a fast metabolism. I'm really good at breaking that food down. But it's not just breaking things down. It is also building them up. So please make sure you circle or star or highlight the fact that metabolism is all of the chemical processes, building up and breaking down. Now, there are two terms associating with this for those chemical processes, and that is anabolic or catabolic reactions. Now, before you start thinking I'm a crazy cat lady, I am going to use another cat reference to help you visualize this. Whenever you're trying to think of the difference between anabolic reactions and catabolic reactions, when you see the word catabolic, I want you to picture a cat scratching apart a sofa, right? Just tearing it apart with their little claws. That is catabolic, right? That is a reaction in which you are breaking down large molecules into small molecules. So we have it in the figure here. Catabolic is breaking down large molecules into small molecules. So again, picture that cat tearing apart a sofa with its little claws. Then anabolic would be the opposite of that. Instead of breaking things down, in anabolic, you're building things up. Okay, so think about anabolic steroids, like think of A for Arnold Schwarzenegger, right? Anabolic steroids, building up muscle. Okay, so remember the difference between anabolic, building things up, versus catabolic, reactions that break things down. Now, any of these reactions can use what we call a catalyst, or in biology, an enzyme. Okay, enzyme and catalyst is the same thing, except that enzymes are specific to biological systems. Okay, these are simply substances that will help reactions, okay, help speed them up by lowering their activation energy. So I want you to circle that they lower activation energy. That will be important. So speaking of why that's important, here we have enzyme action, right? How do enzymes help reactions? And as I just mentioned, enzymes help speed up, meaning quicken, reactions by lowering the activation energy. What this means is activation energy is the energy required to start up a reaction, right? To activate it. So if you lower that activation energy, that means that now that it's lower, you'll get to start up quicker, right? So it's quickening the start of that reaction. It's not getting used up. It's not changing the overall free energy, which would be the reactants versus the products. It's not changing any of that. All an enzyme is doing is helping you quick start that reaction. So kind of think of it as if you have been saving up money to buy a new cell phone, right? Currently costs $1,000. Let's say it's one of the fancy smartphones, full price, expensive. If you have to save up $1,000, that's going to take quite a bit of time. Whereas if they suddenly drop that price to $50, most of you can just go out and get it now. $50 is a lot easier to save up to and to get that ultimate goal versus having to wait and save up to $1,000. So that's what enzymes do. They drop that price. They make it quicker and easier to obtain this reaction. Okay, but they're not doing anything else. Okay, so free energy stays the same, but they lower the activation energy to grab that reaction. The analogy that I like to do with enzyme action, or how an enzyme actually works, the mechanism of enzymes. Let's look at the picture on the next page as we go through this analogy. Okay, so here you have an enzyme, right? This is an enzyme. It has active sites here for substrates, okay? Substrates are simply the building blocks of a molecule, okay? So for instance, amino acids that get put together or interact with each other to make proteins. So now you have this enzyme, and the analogy I like to make with the enzymes and their mechanism is enzymes are like a seat on a school bus, okay? All it's doing is giving two people a place to sit next to each other, putting them in close proximity so that they can interact and start a reaction. So if you picture an enzyme like the seat on a school bus, well, what happens? First day of school, two little kids come in. They sit next to each other on this seat. What happens when you are near each other, especially little kids? What are they going to do? They're going to start chit-chatting, talking, interacting. By the end of that bus ride, they're like, oh my God, we're best friends, right? Because they have now bonded by sitting close to each other, by being in close proximity, okay? That's exactly what enzymes do. They are a seat here for two substrates to sit next to each other in close proximity so that they will then interact and form a bond. And then what happens, just like the two little kids on that seat bus, they get up, they leave, right? They leave, and what happens, that seat is now available for two more kids to sit next to each other and interact and bond. And that's what happens with the enzyme. Once these substrates have bonded and leave, the enzyme has not been changed or used up in any way, and it allows for two other substrates to come along, sit on the enzyme, bond, interact, and then leave, okay? And through that process, if you think about it, by bringing two things in close proximity, okay, they're not actually part of the reaction, they're just facilitating it. They're allowing this reaction to happen quicker because it's forcing two substrates to sit near each other. Whereas if these substrates were, across the room, different parts of a building, they're not as likely to meet up with each other, to bond, okay? So that's what we mean in terms of lowering the activation energy. This enzyme, by having the two substrates right next to each other, they're quickening the start of the reaction. They're lowering the energy required for these two to bond. But again, they are not used up. They're not changing free energy, okay? Now, with this enzyme activity, there are various factors that can affect how well this enzyme is going to work, okay? How these reactions are going to go. Some of the factors include, first of all, substrate concentration. So ask yourself for each of these factors, how would enzyme activity be affected? Okay, make sure for each one you're able to briefly explain how this factor would affect the enzyme activity. So first up, we have substrate concentration. If you have a lot more molecules, right? A lot more substrates present. What will that mean? More molecules or more substrates means more interactions. The substrates are more likely to bounce into each other and bond or interact. So you have increased activity, okay? More substrate increases activity, less substrate decreases activity. The next two factors are related to each other, optimal pH and optimal temperature. These two factors, if you think about it, what did we say enzymes are earlier in this lecture? Enzymes are proteins. What happens to proteins at the wrong pH or temperature? They denature, right? And when you hear denaturing, that means the function is ruined. So if an enzyme does not have the optimal pH or the optimal temperature, it will denature, and this reduces enzyme activity. whereas optimal pH and optimal temperature allows for good enzyme activity. Then we have enzyme cofactors and coenzymes. For cofactors, I want you to circle factor and right above it, write the word factory. I want you to picture a factory, a big building and smoke and metals and minerals, all that stuff, because cofactors are metals or minerals. They are inorganic molecules that help activate enzymes, that help enzymes to work, okay? So cofactors are inorganic metals or minerals that activate enzymes, whereas coenzymes, these are organic vitamins that activate enzymes, okay? So one more time, cofactors are inorganic metals or minerals that activate enzymes, whereas coenzymes are organic vitamins that activate enzymes. Okay, so that's why if you think about even yourself, you always have to have your various minerals that you take in, things like zinc and magnesium, as well as coenzymes, things like vitamins, to help your cells have proper enzyme function. Another thing that can affect enzyme activity is enzyme inhibition. And there are 3 different ones that you have to know the difference between or amongst. Enzyme inhibition can be non-competitive. Okay, in non-competitive inhibition, which you see in this figure here, non-competitive because the inhibitor is not competing with the substrate for the active site. Notice the competitor is binding somewhere else on the enzyme, and that changes the shape of the enzyme, so the substrate can no longer fit. It can't sit down on this enzyme, so it's inhibited. But notice there's nothing actually blocking the active site itself. The inhibitor binds elsewhere. That's why it's non-competitive. It is not competing for the same site. Whereas competitive inhibition over here, you have the inhibitor physically blocking the active site. The inhibitor is competitively blocking the active site, so now the substrate cannot bind. Okay, so you do not get the reaction. The last type of inhibition is feedback inhibition. Okay, when you look at a body or a cell, there is usually a whole bunch of reactions in one chain. Okay, so there's usually multiple enzymes involved to make one ultimate product. And many times this product then goes back and blocks one of the enzymes in that reaction. Because think about it, if you already have that product, why would you want to waste resources, time, energy, making more of that product that you don't need? The cell would just fill up with that product and explode, okay? So instead, that product serves as an inhibitor to block enzymes in that pathway to block production of more product because you already have that product, okay? So you have non-competitive, competitive, and feedback inhibition. Make sure that you're very clear on the differences amongst them. Now I want to point out one other thing is when you hear non-competitive inhibition and you hear a change in shape of an enzyme, you'll usually also hear the word allosteric. Okay, allosteric, A-L-L-O-S-T-E-R-I-C. Allosteric change simply means a change in the shape of the enzyme, and that you find in non-competitive inhibition. Something else we want to mention in terms of metabolism is that a lot of the reactions that we've been talking about, a lot of times we categorize reactions in metabolism as either oxidation or reduction. And you see it written as redox, so reduction and oxidation. A way to remember what exactly is going on is Leo says Gur. So that tells you lose electrons, oxidize, whereas gain electrons, is reduction. Because in redox reactions, electrons are passed from one molecule to another. One of those molecules is losing electrons, and that is called oxidized. It has been oxidized, whereas the molecule that gains electrons has been reduced. Okay, and a way to remember that, if you think about it, gaining an electron, electrons are negatively charged, right? So they're gaining a negative, hence why they've been reduced. You've gained negatives. Now keep in mind that oxidation is taking place at the same time as reduction. So one of the molecules is losing electrons at the same time as one is gaining electrons. The last thing I want to go over with metabolic reactions and metabolism are the energy yielding reactions. Okay? When you look at this slide, there's a few different terms that you have to know. Okay? They're bolded, so you have chemoorganotrophs, chemolithotrophs, phototrophs, and these terms here, heterotrophs versus autotrophs. All of these terms I want you to be very comfortable with. The difference between these terms, first off, is what they are yielding or producing. Heterotrophs and autotrophs refer to organisms trying to obtain carbon. Okay, so heterotrophs and autotrophs is referring to what an organism is breaking down in order to obtain carbon. So heterotrophs means that an organism is breaking down organic matter to obtain carbon, whereas autotrophs means self-feeding. These organisms are breaking down inorganic matter, such as CO2 from the atmosphere, to obtain carbon. And what they do with that carbon is then build the biomolecules that we talked about. Okay, so make sure that you know heterotrophs means that an organism is breaking down organic material to get carbon. Whereas autotrophs, they are breaking down inorganic material to get carbon. Notice both cases, they want carbon. These three terms, on the other hand, these three terms over here, they are breaking things down to get energy. Okay, not carbon, they're breaking things down to get energy. Chemoorganotrophs are breaking down organic molecules to get energy. Chemolithotrophs are breaking down inorganic molecules to get energy. And lastly, phototrophs are breaking down light to get energy. Okay, so notice these three are about energy. These two here are breaking things down to get carbon. Now, the last thing I want to point out on this slide is over here, you see organisms breaking down glucose to get energy. What do we call that? That's respiration or fermentation, right? That is breaking down glucose to get energy. Respiration, which we'll talk about in a minute. Now, in this case, since you have an organism breaking something down, would you consider that reaction catabolic or anabolic? And what you have to do is remember our little visual, right? Catabolic. Scratching, 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 scratching up that sofa, tearing it apart, breaking it down. So whenever you see breaking down glucose, well, that tells you that's catabolic. You are breaking something down. Okay? Once again, you may want to put little stars there and make sure that you know all 5 definitions that we discussed on this slide. Heterotrophs versus autotrophs, and then chemoorganotrophs versus chemolithotrophs versus phototrophs. Okay? Now, the last thing to go over in terms of metabolic processes are the examples of using organic material for energy, which are respiration versus fermentation. Now, if you go over respiration and fermentation in another class, such as biochemistry, you have to memorize all kinds of structures and enzymes and everything. For this course, you don't need that. What I want you to know for these two things is how they will differ in terms of presence or absence of oxygen, and how much ATP will an organism get in respiration versus fermentation, and whether they are a eukaryote or a prokaryote, which we'll talk about more in a couple of slides, but basically the idea is prokaryotes have no nucleus, no, okay, trick, no. Prokaryotes have no nucleus, whereas eukaryotes have a nucleus and internal membranes, and that will affect how much energy they get from this type of reaction. So the first thing that we have here is showing the difference between respiration versus fermentation. Notice that glycolysis is the first step in this breakdown of glucose or organic material. You can even cross out organic compound and just write glucose because we're usually just talking about glucose here. And so notice that glycolysis occurs first, and it doesn't matter whether oxygen's present or not. Either way, glycolysis will occur, and either way, this step of glycolysis will produce 2 ATP, okay, 2 pieces of energy. Then what happens next depends on whether there's oxygen present. If there's no oxygen present, then the organism cannot undergo respiration and instead has to undergo fermentation. And what it does in fermentation is break down the products of glycolysis into lactic acid or ethanol. Okay, and that's why you hear if there's too much fermentation, your muscles, you're building up too much lactic acid. That's not good, right? Get fatigued with that. Now, with this fermentation, what do you notice? How does fermentation over here differ from glycolysis and from respiration? What are you not seeing over here? There is no ATP produced from the fermentation step. Okay, so if there's no oxygen present, you get no ATP from fermentation. So all you get from one glucose is 2 ATP if there is no oxygen present. Because remember, no matter what, glycolysis will happen. So you get 2 ATP no matter what, but then you get nothing extra from fermentation with regards to ATP. What you do get out of fermentation is that the products of fermentation can then go and serve as feeding into more glycolysis, which will give more ATP in that way. Okay? So now, that's if there's no oxygen present. If, however, there is oxygen present, a cell can undergo respiration. And with respiration, what do you notice? Boom, a whole lot of energy, a whole lot of ATP. In this case, up to 36 ATP from respiration. So if oxygen is present, how much ATP can that cell get from one glucose molecule in total? It could get up to 38 ATP because you have one glucose. No matter what, glycolysis occurs, so it gets 2 ATP there. And then if oxygen's present, it gets a whole lot more in respiration, up to 36. So that would be 38 total. So now we're going to look at how the textbooks or online would kind of break this down a little bit more. I kind of made this slide myself to just kind of show you the simple version because, like I said, in this course you don't need crazy details for micro with regard to this process. So let's look at a few more details before we finish up. So, continuing on with the metabolic process of breaking glucose down. We have 4 phases of respiration listed here if oxygen is present. Notice that glycolysis occurs whether oxygen is present or not, and that we call it anaerobic, okay? No oxygen needed. whereas the rest of these processes here are aerobic. They require oxygen. Okay? I want you to be familiar with each of these names of the processes involved in breaking a glucose down. You have glycolysis, prep reaction, citric acid cycle, and the ETC, okay, electron transport chain coupled with chemiosmosis. You do not have to know the crazy mechanisms of each. You don't have to know pyruvate, acetyl-CoA, any of that. All I want you to know is these names associated with respiration. Know that glycolysis does not require oxygen, and note where you're getting your ATP from. Okay, so notice glycolysis gives 2 ATP, citric acid gives 2 ATP, and electron transport chemiosmosis gives 34 ATP. What do you notice? One of these reactions does not give any ATP, and that's the prep reaction. Okay, so make sure to put a little star for yourself and write that the prep reaction is the only phase of respiration that does not give ATP, and it makes sense with its name. Think about if you're cooking, for instance. If you're prepping a meal, if you're cutting up a piece of chicken before cooking it, well, you can't eat it at that moment and get everything that you want from it. You're just prepping it. You can't get the energy from it until you actually cook it and eat it, okay? So prep reaction does not give any ATP. It simply prepares the molecules so that the cell can then break them down for proper ATP in the next steps, okay? So again, be aware, glycolysis, prep reaction, citric acid, and electron transport chain. Make note that glycolysis does not require oxygen, but the other three do. And make note that the prep reaction is the only step in this process that does not yield ATP. And the last thing to note here is how much total ATP can that cell get from one glucose. That total is 38 if oxygen is present. Now make note that that is if we are talking about a prokaryote, meaning something like bacteria. In a couple of slides, you'll get to see that a eukaryotic total is slightly different. Okay, so prokaryotes, if oxygen's present, can get 38 ATP. If oxygen is not present, then they must undergo fermentation instead. And as we mentioned in the last slide, fermentation does not give ATP by itself. Instead, there's only a 2 ATP yield from the glycolysis step. Okay, so make note that there's no oxygen present for fermentation and that the total yield when no oxygen present is just 2 ATP. Okay, and that will be the same whether it's a prokaryote or a eukaryote, because you'll notice that glycolysis takes place in a different spot than respiration in eukaryotes, which we'll talk about in a second. But again, keep in mind, oxygen versus no oxygen, how much ATP we are getting. This is just to show you an example of how you might encounter this process in a figure. Okay, again, this is more complicated than what you have to know. I simply put it here just to help you visualize respiration versus fermentation. But again, you don't have to know all the details in terms of NAD, NADH, any of that stuff. The last thing to note with metabolism is that difference that I kept hinting to with regards to prokaryotes versus eukaryotes. So like I said, the difference between a prokaryote and a eukaryote is that no. Prokaryotes have no nucleus. They have no internal membranes. Okay? What that means is that all of these processes are able to occur in one location, which happens to be the same location where energy can be used in the cell. Everything's mixed together in a bacteria. Eukaryotes, on the other hand, we have internal membranes, right? We have nucleus. We have our various organelles. So in eukaryotes, when they make the ATP, they then have to pay a 2 ATP toll to get that energy across their internal membranes to where they'll actually use the energy. So what that means is eukaryotes will lose 2 ATP from their respiration process to get the energy where it needs to be. Now, what eukaryotes do not ever lose is 2 ATP that they gain from glycolysis, because glycolysis happens in their cytoplasm. There's no membranes to cross over. They get to keep that no matter which process they do. So as a recap, just to kind of go over the different amounts of ATP that cells can have depending on whether oxygen is present or not. If a cell is a prokaryote, if no oxygen is present, then from breaking down 1 glucose, they get 2 ATP, because remember, they get it from glycolysis, and then fermentation gives them no extra energy. In a prokaryote, if oxygen is present, they now get 38 ATP. They get the ATP from glycolysis. They then get it from their citric acid cycle, the electron transport chain as well. Okay? So they get 38, and they don't lose any because they don't have any toll to pay, no internal membranes. Eukaryotes, if oxygen is not present, they get 2 ATP because they get 2 ATP from glycolysis. And like I mentioned, glycolysis, there's no internal membranes there. Okay, it happens in their cytoplasm. So they get to keep that 2 ATP. If, however, oxygen is present in a eukaryote, then from 1 glucose, they would be able to produce 38 ATP, but they would only have a yield of 36 ATP from 1 glucose when oxygen's present, because they have to pay that toll. They have to use energy to get that ATP out of their internal organelles and into the cytoplasm where they want to use it. Okay? So I'll just write that out on the next slide to help you visualize each of those 4 scenarios that you have to know. So make sure to put little stars on them. Okay, so here is the recap that I promised you of how much energy each type of cell can yield from breaking down 1 glucose. So again, a prokaryote, no oxygen, they get 2 ATP. Oxygen, they get to keep 38 ATP. Eukaryotes, on the other hand, no oxygen, 2 ATP, just like a prokaryote would. But if oxygen is present, a eukaryote only gets to keep 36 ATP from breaking down 1 glucose. Okay, and again, that's because they have extra internal membranes. Okay, that is it. You have made it through Lecture 1. Now it's time to head on over to the discussion board, and also please remember to add this lecture to your study guide that you have to prepare. Okay, more information on that is posted on Blackboard. And any questions you have, any concerns, anything you need a little extra help on or would like some practice problems with, please, as always, contact me anytime, day or night, in the Remind app. Thank you and have a wonderful day.